Thank you and good morning, and thank you for the opportunity to tell you about GenVivo, a genetic medicine company that has a lead asset in clinical trials right now. It's our latest stage program, and it's off-the-shelf, intravenously administered, which is an important point. It activates the patient's own immune system, and that's why we call it personalizing, and I'll go into the mechanism in a minute. To seek out and destroy patients' cancer cells with continuously amplifying therapeutic immune responses. We're currently in an expansion phase in hepatocellular breast and melanoma, and we'll be initiating a checkpoint inhibitor combination study this month, actually. It's based on a bicistronic payload that it has a proprietary enzyme and an immunostimulatory cytokine, and we have demonstrated safety and efficacy. Our technology is really based on the gamma retrovector, and so we have a lot of experience in the genetic engineering and production of this particular vector. We call our technology platform the GenVivo, and we think it has some advantages over competitors, particularly in the in vivo CAR T and K spaces, which are our second program, and we intend to be in the clinic next year with that. We're the only company that actually is working on the gamma retrovector in the in vivo CAR T space. We also have a program in IL-12 encoding vectors. Those of you that know about IL-12, very exciting cytokine that does the right kind of things in terms of stimulating the immune repertoire that you like to see in tumor cells, and it actually has anti-tumor activity itself, but it's way too toxic to give IV in terms of the protein. And then importantly, we have our own in-house manufacturing facility, GMP. We work on stable suspension cell lines. A lot's been talked about manufacturing here this week. And we'll show that we're stable, consistent, and cost-effective with a low cost of goods. Our last run through the cost of goods analysis fully loaded was about $200 a vial. So we can produce high-quality product through phase 2. Here's our pipeline, Gen 2 in vivo CAR T, the IL-12 program, and then we have a program in targeted vectors that's discovery, but I won't talk about that today. Here's our timeline. We found the recommended phase 2 dose last month, and so we're in the expansion phase right now with a readout in Q3 of next year. We did an IND amendment to do our combination and intend to dose patients this month. It's a two-stage Simon design, so we'll have a readout fairly quickly. And then we intend to be in the clinic with our in vivo CAR T construct in 2026 as well as the IL-12 program. You know, we're really proud of our GenVivo platform. We think it has a lot of advantages. It has a large capacity. It has surface proteins and envelope that can be modified to target particular desired cells. In this case of the Gen 2, the expression can be regulated by a suicide gene and a prodrug, and that can be used in other cases as well. We integrate for cancer, but we also have a non-integrating vector that would be available for autoimmune vaccines and gene editing. We can deliver it IV, which I mentioned is an important point, also IT if necessary, and I talked about our manufacturing process as well. Let me talk about the mechanism for a minute. It's really a dual mechanism. So when you administer Gen 2 IV, transduces tumor cells. Our vector only transduces dividing cells. Once it's transduced through transcription translation, the enzyme is expressed as a suicide gene in the cytosol, not into the nucleus. When a prodrug's added, you generate a toxic intermediate which causes DNA chain termination, cell death, and release of tumor antigens, including neoantigens, which then in the presence of the cytokine are presented in an enhanced way to the dendritic cells. You have maturation of other antigen-presenting cells, enhanced antigen capturing, and maturation and migration of cytotoxic T cells. As I mentioned before, because we're continually dosing, this is actually an enhancing a process along the way, and it can actually pick up new mutations from the cancers that develop. There's really good synergy with checkpoint inhibitors, and many of you know about CT26 cancer models in mice. It's very aggressive tumor, and you can see here that if we have a checkpoint inhibitor and Gen2, this is the mouse form because of the differences with the cytokine between human and mouse, and you can see that in the presence of a checkpoint inhibitor, we get very, very nice synergy in this particular model, and that's why we're testing that in humans. We have experience with Gen2 in an Asian trial. This was patients that had either a tumor in the liver or because of primary disease or metastatic disease. We looked at several different regimens. The dosing is the first week, the prodrug is the second week, and then a holiday. Very wide dose range across the way. We didn't get the recommended phase 2 dose. That's why we came back to the States. Saw partial responses in HCC patients, stable disease in a lot of patients, and I'd point out that our dosing was over 3 logs. We were concerned about bone marrow toxicity, which we never observed. There was very few treatment emergent adverse events, very well tolerated drug, and at the highest dose levels, we had a median number of cycles that was almost 6, so almost 6 months. We've seen some interesting clinical benefit. This was a hormone response of a positive breast patient with a cutaneous response. She had 3 prior regimens, including classical hormone therapies and paclitaxel carbo. She actually got 17 cycles of Gen2 with a nice cutaneous response. You can see on the bottom there also a response in a cervical lymph node. She had extensive liver disease, and that's why this was stable disease, but actually was on trial, as I mentioned, for over a year. We brought the trial back to the U.S., and it's ongoing right now, both with this intravenous dosing phase, and we're doing intratumoral as a comparison. Our readout is objective response rates, which is obviously what everyone is interested in. And as I said, we'll have data coming soon, both on the monotherapy and the combination trials. To get to the recommended phase 2 dose, 29 patients over 5 different dose levels. We have PK. We have a lot of actually paired biopsies, which is going to be quite interesting in terms of looking at the different effects, particularly immune profile. And we're seeing some interesting pharmacodynamic effects here. This is the first 3 dose levels on the right, the highest dose level, or the next to the highest dose levels on the right, and you can see that we're actually seeing GM-CSF in the serum, suggesting we're getting a response. Here's some of our competitors. They have to deliver their vectors either IT or intravesicular, so locally they either have a suicide gene or GM-CS. We have both. We think we have some advantages there. So these are the advantages of Gen 2. It's off the shelf, captures all the patient's tumor antigens, IV, well tolerated. We have a strong patent position, and as I mentioned, we can make the drug. And we've taken our GenVivo platform and applied it to in vivo CAR T, and as I mentioned, we're the only company that's doing that. Here's some data that's from human PBMCs. We can see that we get really good conduction of CD4 and CD8 cells, and again, these have to be dividing cells, so we're excluding exhausted T cells, for example. We can also show that we have B cell depletion in these human PBMCs, and that's very clear from both the facts on the left and the percentage that's on the right. We can also do this in a lymphoma model, and we're actually in studies in animals right now, and that's data will be coming. So we're very excited about our position in this particular space. We went back and looked at some of the Gen 2 data, and we can see that we actually do see some transduction of T cells in the clinical trial, and we can see that the suicide gene is very effective when it's needed. And so we have that in our system. We're looking at targeting both for transcriptional translational, that means promoters, enhancers, miRNA. We're looking at TCRs and BiTEs. In terms of immune modification, we'll have an armed CAR, and with our experience with IL-12, that's become very helpful. And then, as I said, we have a safety switch if it's needed. Moving on to IL-12, you can see there on the left that it really promotes the kinds of things that you like to see in an anti-tumor therapy. However, IV infusion of the protein is way too toxic from multiple studies on the outside. We have lots of animal data to show that when we deliver our a vector that we have very good anti-tumor activity. We can also show that we have an increase in circulating activator T and NK cells, which you'd like to see. This is a granzyme B, which is a measure of activated CD8 CD4 and NK cells. We can also see it when delivered IT, interestingly enough. In-house manufacturing was talked about a lot here. If you look at the top right, you can see that we've actually increased our titer over 4 log units. In our PR7 process, which is the one that's in the clinic, we've had a 100% response rate. We're a multi-use facility. We use single-use technology, and we have multiple quality systems in place. We'd like to show this slide on the left side, where this was a commercial COVID vector vaccine, the AstraZeneca one. You can see that it was supposed to look like the far left-hand column, but actually looked very messy on the right hand, and patients got 2 times less product depending upon the lot number. We have a very clean preparation that's very reproducible along the way. We have a number of issued patents that are within the space of the Gen 2, the 1013, and active prosecutions, and our others with provisional patents pending. So our key milestones are to establish objective response rate and the duration of response in the third-line HCC and melanoma patients, and disseminated activity in these CPI refractory patients, most of which there'll be. 2-stage Simon design, as I mentioned, to review risk. There's a clear inflection points with the open label readout. In our Gen 310, we want to initiate primate studies very early next year to look for lack of CRS, longevity, specificity, and efficacy, and intend to file an IND in second half of 2026. And in 1013, we want to complete the preclinical testing and file the IND in the second half of 2026. We have an experienced management team. We have lots of experience in oncology trials. Dr. Nori Kashihara, who's in the audience here, just really joined us as our Chief Scientific Officer. He's had many decades of experience in the vector technology. We have a very good scientific advisory board. As you can see, you'll recognize some of the names here with Drs. Ashworth, Cannon, Brennan, Gruber, June, Keim, Koya, Shimlu, and Wong. And so this is our GenVivo's personalizing cancer immunotherapies. We're going to capitalize on the synergy between Gen 2 and the checkpoint inhibitors in the area of unmet medical need with systemic administration and off-the-shelf therapy. And we have a 2-stage Simon design, and sort of really show that we have these the ability to look at the continuous amplification of therapeutic immune responses with all of the studies that we have planned to look at that particular readout. We're very excited about our in vivo CAR T intervention with specificity for T and NK cells. We think that the endogenous properties of the vector are very much in our favor, and our ability to do genetic engineering and manipulate very quickly is also to our advantage, along with the fact that we can manufacture in-house and speed things along. In terms of Gen 13, we're very interested in really harnessing the power of IL-12. There's other programs that are out in the clinic, and we believe we can be very effective in that regard. And also, we've used the knowledge we've gained from IL-12 animal studies to help shape what we're doing in terms of our armed CAR T. And so we know that we're behind, and so we want to really generate the optimal product candidate going forward. Our GenVivo platform can be applied to other indications, autoimmune diseases, vaccine, and gene editing. It's there to take advantage of, but we're trying to stay focused on the programs we have in-house. And as I mentioned, our in-house GMP manufacturing facility, our people are really good, and we've generated high-quality, consistent, cost-effective product candidates, and we have a strong composition of matter patents in all our important programs. Looks like my time's all up, so I'll be happy to take questions in the rear. Thank you very much.